Everyone. So thank you very much, everyone, for coming to our innovative discovery series. We are really delighted today to have Dr. Druley, who is going to talk to us about rare DNA and detection of rare DNA and RNA mutations, and Dr. Krauge, Erin Krauge, who is here from Nemours, who is Associate Director of Bioinformatics at Nemours, will introduce Dr. Druley. So before introduction and the talk, I have some housekeeping points that I need to say. So for CME credits, you must sign in and must include your email and credentials. The sign-in sheet is in the back. Next week, we take a break for Memorial Day, and we'll return with a tech talk on June 1st on identifying optimal cut points for continuous predictors to discriminate disease outcomes by Beth Wolf from MUSC. And this will be followed by three weeks of ID series. The first one will be Advances in Genomics and Sequencing by Renata Pellegrino from CHOP. That will be on June 9th. Then Analysis of Interview Data by Kathleen Breyer-Smith from University of Delaware on June 16th. And Big Data to Knowledge by Kathy Wu from University of Delaware on June 23. So please remember all this, and you can sign in on our website for our Innovative Discovery Series, and the full schedule is posted on our website. So thank you very much, Erin.
So thank you so much for coming out today. So it's my privilege and honor to be introducing Dr. Todd Druley today. So for those of you who don't know, Todd recently was awarded an R01, and myself and Angie are collaborators. We work with him on this exciting project, which you're going to be hearing a lot about today. His major focus is in pediatric cancers, but yesterday I got to witness him save a grown-up man's life in real time, right in front of Nemours. So he has quite the extensive array of knowledge and ability to impact people's lives, so it's pretty impressive. So I'll go ahead and let Todd talk because I think he'll do much better.
All right, thanks everybody for coming and the invitation to be here. Thanks to those of you who have dialed in. So I was going to talk a little bit today about some technology that Erin has helped my lab and analyze stuff that we've developed to try and improve molecular diagnostics aimed at pediatric cancer, because that's what I do, but it's not exclusive to pediatric cancer. It's actually not even exclusive to cancer. So I'll show you a little bit here. So I don't have anything that I need to disclose. I wish I did, but I don't. So we'll go right ahead. So I want to talk about clonal hematopoiesis. And so we've started using this technology, and I'll explain that in a minute, to look for residual leukemia after a child's been treated. This is known as minimal residual disease, and I'll talk about that a bit more. But we've started applying this to adults because that's where the most information is known about adult clonal hematopoiesis during myeloid leukemia. And then we've since taken this back to umbilical cord blood. We've got an application in Down syndrome AML, and then all of the work there in point number one is all DNA based, and there's a lot of information. There's a lot of information that can be gained from RNA, and so we don't want to be blind to what's happening in the RNA space. I think there's recent data showing that a lot of the children are more likely to have cryptic fusions than point mutations, at least relatively compared to adults. So we wanted to build things to do that as well. So starting with error-corrected sequencing, or ECS. So let's start here with the clonal hematopoiesis in adulthood. So there's multiple ones of these studies. This is just one that was published about 3 years ago by the lab of Ben Ebert in Harvard. And so he recognized that they had a population of about 17,000 people with diabetes, and those individuals had had their blood samples sequenced and looking for genetic variation, mutations, polymorphisms that could be attributed to their metabolic syndrome, but his group recognized that they might also have clonal mutations that could be associated with myeloid leukemias and AMLs in adulthood. So these people weren't selected for having leukemia, but they just had this huge data set. So I've put an arrow here at this 60 to 70 age range where they reported that there's about 5 to 10% of individuals that they could find subclones in the blood sample that had mutations in genes that were associated with myeloid leukemia. And the most common one shown here is DNMT3A, which we know very well is frequently found in adult de novo AML. And then there was a tail of other genes, TET2, ASXL1, P53. So Ben and other folks have named this CHIP for clonal hematopoiesis of indeterminate potential. And so that was pretty rigidly defined as having to be greater than or equal to 2% variant allele fraction. And so there was the ability to track some of these individuals in this study and see that there's a somewhat increased risk of developing AML if you had these CHIP mutations at an earlier age. So, but there's nothing biologically magical about 2%. It's really based on the limit of detection of the sequencing platform, which I'll explain in just a moment. So now back to pediatric leukemia and minimal residual disease. We have great success with lymphoid leukemias because you have a clonal T or B cell surface receptor. This was work published a few years ago by Mike Loken, working with the Children's Oncology Group. And so he has spent decades really developing profiles of normal hematopoietic clonal population development. So if he sees a child who's known to have a myeloid leukemia and he sees a subpopulation that has a different profile of surface immunophenotypes, he is able to attribute that to residual disease because there's no single clonal epitope on a myeloid leukemia cell. So you don't have the same level of sensitivity. For ALL, you have a level of detection of approximately 1 in 10,000. Right now, it's 1 in 1,000, maybe 1 in 2,000 or so right now for most of the AML stuff. So clearly, it's better. Here's children that have residual disease, and their outcomes are significantly worse than children that don't have residual disease. But we've got a third of the children here that don't have any residual disease by the flow cytometry metrics and still suffer inferior outcomes. And one of the reasons that is because it's increasingly recognized that from the time of diagnosis after therapy, a month or two in, the profile of surface immunophenotypes on those myeloid blasts can change, and so you get a false negative. So this is a binary output. Yes, you have residual disease. No, you don't. So what if we could introduce a sequencing platform that would move us out of that binary yes or no phase and give us more gene-based data from which we could use to inform our therapies for children that have difficult-to-treat leukemias? So the reason that sequencing has not really taken off as a really strong tool is because of the relatively high error rate of next-generation sequencing platforms. So what I'm showing you here are 2 known mutations in P53. They're both circled in blue. And this shows you that all of these other black dots in both of these samples here are just noise introduced by the sequencing platform. So the noise threshold is, depending on how you do it, 0.5 to 1%. So you can think back, if we define CHIP as being greater than 2%, that is simply because the odds of that very rare event being noise introduced by the sequencing platform above these 2% thresholds is much, much less. But below 2%, it's going to be difficult to distinguish true variation from noise. So that's really the technical limitation with next-generation sequencing. And you'll hear people talk about doing deep sequencing. So that means instead of covering your target region 30, 40, 50 times, you want to cover it 250, 500, 1,000 times. That's great. That helps you determine a population in your sample that's maybe 10% frequency, maybe down to 5% frequency. But you can't beat the error rate, excuse me, of the sequencing platform by just sequencing more. You're still generating noise at the same rate. So our error-corrected sequencing approach that I'll explain in just a moment is not particularly unique. I mean, there's multiple labs and companies that are trying to now introduce these types of strategies. There's a few different things in the DNA space. A lot of people like duplex sequencing. There's other products in the RNA space that do similar types of things. So I don't want to act like we're the only show in town. I think we've been able to develop a workflow that's very easy. It melds right into the Illumina workflow. So if you know Illumina sequencing, it's pretty seamless with that process. So we're really trying to build a toolbox so that we could provide anybody with a more precision or personalized approach to their molecular diagnostics. And so what can we find in each of these spaces? Point mutations, small indels, inversions. We can, in the RNA, we're looking for cryptic fusions, allele-specific expression. We can do more quantification of very low frequency transcripts, things like that. So how this is done is pretty simple. We've used the mutations in the cancer as a biomarker. And this was really worked out as the thesis of a former student of mine, Andrew Young, who's now back in medical school. And so what we want to do is add this unique molecular index, UMI, to every single DNA molecule. So we're using a 16 base pair random sequence. Doesn't have to be 16 bases. You can make them longer or shorter. And so every DNA molecule gets a unique identifier. And so as you have this cancer-derived molecule has a mutation, that mutation at that position will always be, sorry about that popping up there, but whoops, that will always be associated with the same index. So the index and the mutation are always associated. The noise, these yellow boxes, keeps, it will pop up at random throughout these reads. And so when you computationally aggregate the index, you can say, well, the yellow spots, the noise, only shows up sporadically at different positions. I'm going to discard all of that. I'm going to retain changes from the reference sequence that are in every single read at the same position and always associated with the same index. And so then what I showed you before now turns into this. So here's what I showed you originally. Then when we apply error correction to that, we can drop the noise out, and these mutations that were previously indistinguishable from noise are now readily distinguished from that background. But we want to have something that goes 1 to 1,000, 1 to 10,000, so I'm only at 0.5% here. So we've done a lot of serial dilutions to see what our limit of detection is. So we've looked at GATA1s on the X chromosome, so we have a dosage issue there. I think we're doing pretty good. We've looked at small insertions and deletions, and that holds up well. There's a couple autosomal genes with known point mutations. So we feel like we're confident in our ability to find mutations to 1 in 10,000. We validate our results by droplet digital PCR. And so, now we're in the range where we could use this as a tool for residual disease following chemotherapy, looking for something as rare as 1 in 1,000, 1 to 10,000, and see if we can match flow cytometry. So we've published this a few times. There's a paper from a couple years ago that we published with our adult colleagues. We were able to show that in adults who get therapy-related AML, many of them have P53 mutations. So the question of this project was the P53 mutation introduced by the chemotherapy for the primary malignancy, or was it already in an HSC, a hematopoietic stem cell, and simply selected by the chemotherapy? So we were able to demonstrate with ECS that is actually the selection of a pre-existing hematopoietic stem cell progenitor with the P53 mutation, which led to the therapy-related AML. This paper is more of a methods type paper, just describing how this technique is done. All the sequences to do this are published. Everything's available. We don't have any any secrets that we're hiding. So it's all out there. Now that was tracking things that we know, but we want to be able to discover new things and build clonal architecture for any individual at any point in their therapy, at the time of diagnosis, after treatment, at relapse, if they happen to relapse, a year later, what have you. So we need a bigger panel. We have to be able to survey a lot more gene space and not just go tracking things that we know. And so we could have built our own panel, but we chose to work with Illumina because they sold this nice myeloid sequencing panel. And so it didn't offer error correction. We were able to add error correction to this pre-existing product. And so what it offers is sequencing of 141,000 bases across 568 amplicons from these 54 genes, which are the most commonly mutated genes in adult AML. And so with that, the first thing we needed to do was really, if we're using a tool that's 100 times more sensitive than what people have used previously, we need to know what normal looks like. So to do that, we were very fortunate to be able to collaborate with the Nurses' Health Study out of Harvard. So this is a study that has about 125,000 nursing participants. It started in the mid-1970s. The reason they chose nurses was that the nurses were able to draw their own blood, and they provide a blood sample every 10 years, as long as they're willing to participate. So they've got 40 plus years of blood samples from over 100,000 nurses. Now the study we really wanted to do was comparing healthy nurses to nurses that got AML. They said the AML samples are pretty precious, so prove to us with healthy nurses that you know what you're doing. And so that was this paper, which was published last July. And so we got 40 samples from 20 nurses. The first sample was banked at an average age of 57, the second sample at 68. And we applied this panel of error-corrected sequencing across all of these genes to see if we could define what normal clonal hematopoiesis looked like in the 50s and 60s. As you remember back to the first slide, we are expecting to see, that there's 5 to 10% of individuals that have clonal hematopoiesis in these AML genes. So this is what we found. 95% of those nurses had a validated clonal mutation in one or more of those 54 genes that are associated with AML. But they did not get AML at last check, so there's 20 or 30 years of follow-up after some of these samples were provided that the Nurses' Health Study is confident they didn't get AML. So the median age, so the 57 years and then 68 years. So compared to the paper by Ben Ebert, which found 5.7% of individuals, and then a couple other papers roughly the same, we were able to identify that pretty much and we're only looking at 54 genes. So if you looked at 100 genes or 500 genes or 1,000 genes, there's a lot of variability going on. So, so this is happening to everybody, I think, as a normal product of aging. So I'm not trying to be an alarmist here. I'm just telling you what we found. We published this. But then it leads to the next question, which I'll get to in a second after this. So what I expected to see in our data was that we're looking at such rare events that we're going to see the stochastic random mutation rate across all the genes. And there's not going to be real biology there other than just this is the random noise in hematopoietic stem cell compartment. And that's not at all what we saw. We saw a profile of gene-specific variation that matched what they found with much less sensitive sequencing. So overwhelmingly, the most common mutations are in DNMT3A, TET2, ASXL1 here is the fourth most common. So we see almost identical profile using this very, very sensitive tool. Not at all what I expected to see. And quite honestly, I don't have an answer for why DNMT3A is so much more mutated than other genes. This is not a hot spot at one base or 1,000 base pairs. This is across the entire open reading frame. That's a great question. Let me get to that in a second. All right, I'll show you the data because we actually were able to test people that got AML. So that's the punchline. So we validated this with droplet digital PCR. So this is not a sequencing-based approach. And across 3 orders of magnitude here, I think we did pretty well. So the mutations that we're calling by sequencing, we're finding at the same frequency by ddPCR, even at very, very low levels. So the next thought was that why are these DNMT3A mutations so common? They must be providing some positive selection. So if that were true, then the mutations we find at age 57 should be much more abundant at age 68. And so what I'm showing you in this top panel here is 4 samples. So we did technical replicates at time point 1 and time point 2. Each box is a different individual, and so each line is a different mutation. So you can see that at median age of 57 compared to 68, there's very little, if any, proliferation or expansion of that particular clone over that 10 or 11 year period. So these mutations don't seem to be allowing those progenitors to expand. So then we thought, well, maybe they're not actually progenitors. Maybe they're long-lived B or T cells that have just been circulating or sitting, fairly idle over all that time. So we were able to flow sort these frozen samples, and the best we could do, given the age of the samples, is just BT and myeloid, and then we could do droplet digital PCR in each of those compartments and see what we found. So we were able to find those mutations in all 3 compartments at approximately similar frequencies to the bulk sample. And so that says that the mutations are not happening in a terminally differentiated lymphoid cell. They're actually happening in a progenitor that goes on to make multiple lineages of blood cells. So that leads to the next question, and the Nurses Health Study finally trusted us enough to give us the samples that we originally asked for. So if everyone has these rare clones in genes that are associated with AML, why do so few people actually get AML? So here's what we've done. So we've added to the Nurses Health Study the Health Professionals Follow-Up Study. So this is a study that includes all men, and it was started about 10 years after the Nurses Health Study, which is all women. So they found individuals that did go on to get AML years after their blood samples were collected, and they matched 2 controls for every one of those individuals, matched by age, gender, and ethnicity. We were blinded, so we just received the cell pellets. They were de-identified. We had no idea who got what. And so we could use error-corrected sequencing to look at the longitudinal mutational profiles and see if we could identify who got AML and who didn't. Now, one thing I want to point out, if the answer is because there's multiple hits in one cell, our technology is blind to that. So you could see a healthy person have 8 or 10 mutations and a person with AML have 8 or 10 mutations. But the person with AML has 3 or 4 or 5 of those in the same clone. We wouldn't be able to detect that. And whereas the healthy person has 10 or different clones. And so if there's a single cell burden, we're not going to see that. So here's just one snapshot of these data. So you can see just qualitatively looking here, the cases, the people that got AML compared to the controls, the amount of detected mutation is pretty similar. There's not big differences here. That's the only thing to appreciate from this graph, I think. Maybe a little bit less synonymous hits in the cases, but overall it looks pretty comparable. And so we validated with that with droplet digital PCR, and again, I think we're making accurate base calls. And so the punchline that you asked me a bit ago, so what does associate with actually getting AML? And it was these 3 amino acids, DNMT3A R882, which I'm sure you may know of if you treat AML, ASXL1 1183, and JAK2 V617. So these turned out to be predictive for developing AML with a frequency as low as 1 molecule in 5,000 up to 15 years in advance of actually being diagnosed with AML. Now, this is a small sample size, so our confidence intervals are going to be large. There's no way to get around that. The p-values are pretty good, but I think the odds ratios are what really stand out to me. I think you see a lot of studies with these sample sizes, and the odds ratios are 1.7, 2.1, things like that. This is, odds ratios of 5 to 10. These are very strong effects imparted by these bases. So why DNMT3A R80D2? So in this, so we only got one sample from the men. We didn't have 2 time points for the men. So I can't say anything about that. The women, we were able to get 2 samples from. There was no difference between the first sample and the second sample. So the 882 mutation has been published to be a dominant negative of DNMT3A function. So the protein has to homodimerize and sometimes tetramerize to be able to exert its methylation function. And so when the 882 mutation, either the H or C, is there, it can dimerize, but it can't have its function on the DNA methylation. And whereas if you get a mutation elsewhere in DNMT3A, it either doesn't stop the protein complex from DNA methylation, or it just doesn't bind to the wild type protein, so it's essentially inert. JAK2 V617 is very common it's more common in men than women, and we only found that in men. Interestingly enough, I don't know why. That's more common in men than women, but that's been published before. And it's a frequent pre-leukemic mutation in myeloproliferative neoplasms and myelodysplastic syndromes, so it raises the question as to whether these gentlemen had some prodrome prior to developing AML. And so they didn't have de novo AML, they actually had what would be called secondary AML. At either rate, it's still predictive, but so we're working with Harvard to try to tease that out from their records a little bit more. So to me, I hear a lot of people say, well, we sequenced the exome, we didn't find what we want, we should sequence the genome. Now we sequenced the genome, we didn't find what we want, let's sequence the transcriptome. We sequenced the transcriptome, we didn't find what we want, let's sequence the methylome. And it's this sort of, running on the hamster wheel to try and generate more and more and more data that we know less and less and less about. And I think, these data suggest that if we knew the right things to look for, we don't need to just keep adding layers and layers and layers of massive data sets. We need to really focus on what's associated with the disease. I don't think these are drivers. I think these establish a pre-leukemic state. So we're all getting DNMT3A mutations. And there's, Bert Vogelstein got a little bit of heat a couple years back for his sort of random bad luck paper, but if you have the unfortunate incidence of having a clone with an 882 mutation, this would suggest that, you have to be very cautious for the development of AML. Whereas you're mutating DNMT3A elsewhere, it's probably pretty biologically insignificant. So that's where we're at on that project. Now, I think I told you I'm a pediatric oncologist, so I want to figure out what's going on in children. The clonal hematopoietic profile in adults was very different from what we expected to see. So we don't really know, I think, what's going on in children. We know there's a big difference in the genes associated with pediatric leukemias compared to adult leukemias. So I think one of the first things we've tried to do in my lab, we're definitely going to sequence a lot of leukemia samples, and that's where we want to go, but we have to understand what the profile of normal looks like first. So one of my students is very interested in this data. So the incidence rate of leukemia is quite low, but after getting an umbilical cord blood transplant, the incidence of leukemia, not a relapse of the original leukemia, but a leukemia that is different from your original leukemia, is about an order of magnitude more than that. And so when my student looked at this, his name's Wing, when Wing looked at this, he thought maybe there's clones in the umbilical cord blood that are being selected through the transplant process that are then lending themselves to an increased risk of a second leukemia later. So we've started to perform error-corrected sequencing on just random umbilical cord blood samples to see what we find. And so the punchline here is similar to what I showed you in adults, is that we found the same 3 papers had looked at clonal hematopoiesis in people less than 40 years of age and found roughly 1% of those samples had clonal hematopoiesis. So far to date, we find and have validated that 18% of the cords we've looked at have validated clonal hematopoiesis. Not in adult AML genes, but we've added, I think I show you here in a second, we've added 40 genes that have been published to be frequently mutated in pediatric AML. So there's no DNMT3A in any of this. That's not seen in pediatric leukemias, and we haven't found any of that here. So one application that I wanted to show you that we are looking at was in Down syndrome AML. So if you're not familiar, Down syndrome AML is heavily associated with the GATA1 gene. So GATA1 is on the X chromosome. It's not on chromosome 21, but almost every single case of transient myeloid transient myeloproliferative disorder shortly after birth or AML in children with Down syndrome is associated with mutations in exon 2, some in exon 3 of GATA1. So we don't need to sequence a big panel to be able to find mutations in these 2 exons. What we did was just develop 2 amplicons. This one spans exon 2. This one spans the 5' region of exon 3. Sorry, this keeps popping up. And so we can look in Down syndrome AML cases and see what we find. So this is one example of a little girl with Down syndrome, and we found a 4 base pair insertion that had about a 30% frequency. So this is at the time of diagnosis, so roughly 30% of the cells were leukemia cells. And you can see that here. This is that blue mark. So if you haven't seen these types of data, this is your chromosome here, and these are the actual reads that are sequenced, and then we align them back to this reference sequence. And so all these little other dots you see are noise, basically, but you see the same blue sequence show up. So that's your 4 base pair insertion. So then after a month of treatment, the same little girl gets sequenced, and that blue box is gone. So we didn't find any evidence of that insertion in her sequencing or the droplet digital PCR. So it's very favorable for her that we've cleared her leukemia, and she's in a complete remission. This is what the droplet digital PCR data look like. So you have 2 fluorophores. One is for wild type, one is for your mutation of choice. And you're making liquid aqueous droplets in an oil background. So ideally, you're going to have 1 DNA molecule in every aqueous droplet, and all the reagents for PCR are in the solution. And so as you amplify that molecule in isolation in its own aqueous droplet, you'll either get the wild type fluorophore or you'll get the mutant fluorophore, depending on what's in there. And these are a lot of droplets that didn't get any DNA inserted in them, so they're just empty aqueous droplets. And so here you can see, this isn't the same case I showed you before, but we have an error-corrected sequencing frequency of 1.3%, and this is 1.7, 1.8% of all the droplets that were analyzed in this experiment. So it tells us that our sequencing frequency was very, very close. In this same child, after a month of treatment, we couldn't find any of the mutated sequences. So another case of a complete remission. So I don't expect you to read all this, but I just wanted to point out some of the data of where we're at. I think this is still in development, but overall it looks pretty good. 83% of the cases we've looked at, we can identify what the GATA1 mutation is. There could, why are 17% of the cases not revealing something? There's multiple reasons why that could be that I've listed here. But, if you look at this case, 75% at diagnosis, there's still 5% after a month of therapy. So you have to worry about that child. Here's a child, 21%, and then less than 0.3. Some of these have cleared. We've got a clone at 5.3% and nothing at end of induction 1. So we're just starting to generate numbers where we can look at the impact of clonal hematopoiesis as a tool for residual disease detection. Any questions on that? I'll switch back here to using single molecule discrimination, less so for DNA and more for RNA events to finish up here. The significance, we're simply using it as a biomarker. I don't. In exon 3? In GATA1? So, I mean, there's point mutations here. I mean, here's an insertion. So it's pretty variable. I mean, the children all have different things, but whatever mutation they have seems to have some defect on GATA1 function. I don't know that it's been carefully discerned what the mutations do in exon 2 versus exon 3, but you know, we're just trying to use them as biomarkers. Yeah. Yeah, that's a great question. So, you know, most of the time, yes. You know, so, but as of right now, in the prospective set, we're blinded to the clinical outcomes until the study's completely finished. We don't, so we're not allowed to be biased. So this is some retrospective samples from a prior study, and you can see the data here is somewhat clear. You know, here's 18%, and this child was known, you know, this is from several years ago, so this child went into remission and stayed leukemia-free. You know, some of these others, here's 8.7 and nothing. But then some of these others are very, very low. So the question is, are we actually detecting the leukemia at diagnosis, or are we not looking at the right space? You know, so we're going back. But you know, in the few samples that we've been able to know the outcomes, and we have data that we believe, I think you know we're accurate. But there's definitely more development that needs to. I think we need more than just 2 amplicons. I think we need to tile across that space a little more comprehensively. And you know we're part of this prospective study that's going to be going on for the next year or 2. So we'll be able to get a few hundred, we hope, of these cases for analysis. Yeah, this is from a published paper. So this is from a paper in 2011. So I'm just giving you a map of kind of where GATA1 mutations seem to occur in Down syndrome. These are not from our data. So the top is transient myeloproliferative disease. So a lot of these Down syndrome children are born with what looks like a myeloid leukemia, but it usually resolves spontaneously. About 25 or 30% of those children will go on and have full-blown leukemia by the age of 4. So this is showing the mutations in that transient disease after birth, and these are the mutations that are being described in the full-blown leukemia. And several children that get leukemia didn't have any transient myeloproliferative disease. So those are good questions. Anything else before I move on to the RNA stuff? DNA. So this is really looking for structural variation, whether it's in DNA or RNA. So everything I've showed you up to now, the magic here is that we're forcing that to align into a preconceived reference sequence. And so when you have a structural change, it's very difficult to see unless you're doing very creative bioinformatics, because the structure is going to be very different from what your preconceived reference sequence is. So for instance, the internal tandem duplication of FLT3 is a common event. There's probably 20% of children, and maybe adults as well, have some structural variation in this gene. And so if you have insertions of 300 to 400 base pairs and you're trying to fit that to a very specific reference sequence, you can't force a square peg into a round hole. So one way that Aaron has been working to improve our ability to detect this is with a company called Archer. And so I think the beauty of this strategy is that we're not forcing the data to align to a preconceived reference sequence. We do what's called de novo assembly. So we generate read lengths of different sizes, and then we can align them to each other. Erin's doing this. I'm not doing it. But she aligns them to each other. And then you can get very, very precise, because every molecule has its own index. We have that UMI on every molecule as well. Now it's on, RNA or DNA. And so we can see here that 71.7% of the sequencing data that we generated shows us that there's a duplication in exon 14. And it starts here in intron 13, whatever that is there. But now if we go to the bottom, we can see we've got a bigger segment of intron 13 that started somewhere else, but we see the exact same duplication of exon 14 in another 10.9% of our sequencing data. And then to bring us to 100%, 17.4 here is just wild type. There is no duplication. So we're able to identify very, very precise molecule sizes and fragments, and I think we do better. One, because we have the UMI approach, and two, because we're doing de novo assembly, which doesn't always work. If we did pretty good with the small indels, the 4, 5, 6 base ones. So you don't need to do this if you're finding something very small, but you certainly do if you're looking for bigger structural variation. This didn't come across because I converted this to PDF, but the point here is that we've got 14 cases. These are all from Nemours, and so this shows their clinical data from standard cytogenetics and clinical testing. And so most of the ones that were described were confirmed when we did the more sensitive approach. 1, 2, 3, 4, 5 cases confirmed, 6 actually. We didn't find anything here. But then the ones that have asterisks here, 50% of these cases didn't find a cryptic fusion event, which Erin was able to identify in her analysis. So 50% of the time we can identify a leukemia-associated cryptic fusion that was not identifiable by standard cytogenetics. Yeah, that's a great point. So, you've missed the opportunity to customize therapy or augment therapy in an appropriate fashion. And another phenomenon that Erin has noted in her analyses is that a lot of times, mutations that are not coding get discarded because it's not always clear what to make of those. So here's one individual, but there's certainly others that have splice site mutations. So she's seeing that when you have that digital readout of the mRNA from somebody who's got a splice site mutation, you're not getting one mRNA molecule. Especially here in RUNX1, she's finding a lot of different mRNA molecules from an individual who doesn't have a coding RUNX1 mutation. So here we've got exon 2, duplication of exon 4, and then exon 5. The same individual has another quarter of their data where they've skipped exon 5 altogether. This is an interesting phenomenon that I don't think has been very well described before, and we're trying to quantify, and she's doing a great job digging through that data. So when you start to compile all this and say, what can I do across a cohort of individuals or a bigger population, what I'm showing you here is Erin's analysis looking at expression values and sort of modified heat maps. So each box is a different gene. And then at the bottom across here, we have a different isoform of those genes. So for instance, these 3 just have a single isoform. There's several isoforms for some of these others. And every row is a different individual in these data. So right away, you can start to pick out the individuals. Here's a person who has overexpression of 2 different isoforms of NUP98. I put an arrow here. There's a couple individuals that have overexpression of this gene. Perhaps this isoform should be the dominant isoform. I don't know the biological significance of these, but you can see there's 3 individuals very clearly that are lacking expression of this. And so, here's overexpression of retinoic acid receptors. So you can really, I think, build these types of models to see what's happening at a very precise level. And so then taking it even further, Aaron's been able to do these pathway analyses. So again, I like that this argues against just endlessly generating data that you're not quite sure what to do with. We don't need to sequence every gene in these clusters. We need to sequence some critical nodes. So if there's an agent that's known to be effective within this mechanistic pathway, and we have data suggesting that the pathway is dysfunctional in some way, it gives us a hypothesis of what we might test, either in a patient-derived xenograft model or a preclinical model for a larger study, something of that nature. And then you can actually start to segregate the individuals using broader data sets. So, there's no question boys and girls have different outcomes and need subtly different therapies, and I think that's probably true for ethnicities, and it's certainly true for ages. And so if we start to have intelligent models to build all of that, we could improve risk stratification significantly as we use this as a prognostic predictive tool. So in summary, just point out that the UMI has overcome the error rate of next generation sequencing. I think at all ages, we're finding that clonal hematopoiesis is more common and quite more variable than what was previously thought. I think, so we're working on this paper. There's really 3 residues, but 4 different mutations that associate with a significant risk of adult AML years in advance. And then we can also use the same UMI approach, although find different data outputs with DNA and RNA to really build a more comprehensive risk stratification profile. So in conclusion here, where are we going next with this? So with Aaron and Andy's help, as his role as the director of the Myeloid disease program within the Children's Oncology Group, we've been able to build the study that Aaron mentioned. It's already gotten R01 funding, and we've been through all the red tape to try and get access to about 2,000 samples from the biggest phase 3 de novo pediatric AML study that's been completed in the United States thus far. And so when we have 1,900 samples from 870 children, and we're going to apply error-corrected DNA and RNA sequencing to all of those samples and compare that to existing flow cytometry. So the point here is to try to improve MRD after the first month of therapy, but I think we really have an opportunity to more precisely characterize clonal architecture at the time of diagnosis and for children who relapse, because presumably the relapse is selection of a minor clone or subclone within the larger data or the larger disease set at the time of diagnosis. So that's great that you can augment therapy after a month shows you that there's still residual disease, but could you add a particular agent from day one if you knew that they had a clone in there that was going to be resistant to standard therapy? So that's where I think we'd like to go. So thank you very much for your attention and the invitation. I want to thank Erin for her hard work in this and Andy for his support and guidance. Lots of folks in my lab that work very hard, have done a great job, and several people at Archer and Illumina, and definitely want to point out the Leukemia Research Foundation of Delaware. They've been a big supporter of Aaron's efforts and overall helping to make this work possible. So thank you, and I'll take any questions. Yeah. So I've got a whole other talk on infant leukemia. My clinical interest is in pediatric cancer predisposition, and I'm really mechanistically interested in how mechanisms of normal childhood development can go awry and sort of predispose or make an environment for early childhood cancers. So under the umbrella that you're describing, we find some information in infant leukemia, which occurs in utero, has terrible outcomes. And it's not just because they're newborn babies, it's because the disease is somehow fundamentally different in a way we don't understand. So it seems as if there's a precise germline genetic environment. So these stochastic mutations that are happening at random in the population, only the people where that falls on that right background that's one of our hypotheses. There's certain individuals that have the right germline environment that would predispose to transformation if one of those events sort of falls on it. And so, pulling together the germline, and then you've got this very rare mutation rate in hematopoietic progenitors, or any cell. I've got a neurosurgery colleague who wants to start doing 4 or 5 different biopsies of brain tumors, and then using error-corrected sequencing to try and build a geographic map of clonal variability with a solid tumor. So that's something we're starting to explore. I think it's exceptionally complex, but it's also quite subtle in many ways. And I think there's a lot we can learn about normal developmental biology, any age really, and then how cancer sort of hijacks those systems based on age, gender, AYA medicine, adolescent young adult medicine, is really sort of the third tier. We think of pediatric and adult, but it's really pediatric, adolescent, young adult, and I think older adults. Their cancers are different, the genetics are different, and require different strategies to treat and maintain.
Are the mutations that you found, are they additive? Is there
Right, yeah.
potentially?
So the question is, are the hits additive? They're either additive or epistatic. I don't think that these are transformative by themselves. But since we're currently blind with this strategy to the co-localization of mutations within the same cellular background, we can only make loose inferences. So I'm actually starting to work with a startup in California who's built a box for single-cell DNA sequencing, and we're going to be one of the beta test sites, one of 3 beta test sites in the country, so that we can take the nurses' health study data that we already have, compare nurses that we know got AML, and we know what their mutational profile is versus nurses that didn't, and then see how much co-localization is there within a single cell for these very rare events. And I think that will speak a lot to the additive or epistatic nature of it. It's definitely moving towards a single cell approach. We've stayed away from single cell transcriptomes up till now because the current methods are only able to survey the top 10 to 20% of transcripts. So the very rare transcripts within a single cell are still not seen, and I'm not sure that we would be able to really do a fully comprehensive survey if we were missing 75% of those.
It's completely, but why not use error-corrected sequencing for everything? If you're looking at rare diseases that population, where there may be rare variants that are associated with disease. IR sensitivity.
So this is filed under patent number so I like that idea.
I mean, is there any reason not to? Is it too expensive? Is it does it take too much intensive bioinformatics?
I think the reason it's not used more right now is more conceptual than technological. We really built this to just plug and play with the Illumina system. strategies use crazy molecular biology and completely disparate workflows. They work great, but they are elaborate protocols that are time consuming, and a lot of the data generated actually at the discard. So, we've created a system that we're very happy with. So, case in point there is that we only get one strand. So we have a top strand, a bottom strand. We only test one strand, top or bottom. So people like the idea of being able to test the top and the bottom strand simultaneously. So there's protocols to kind of put them together and then sequence them in one set. And it's great when it works, but people that are doing it say only about a third, 25 to 33% of the data actually works. So I think there's a lot of expense by throwing out 2/3 of your data. So, another big limitation is that we don't know what we don't know. So we're looking at a small set of genes that we know are specific for leukemia. Not sure we actually know all the pediatric leukemia-specific genes yet. I'm sure there's more that need to be identified. But you would need to have a gene-specific set for glioma versus a medulloblastoma versus an astrocytoma versus a hepatoblastoma. And so you really want to customize it. We can't do this across the whole exome data set with these populations.
There's also a bias with the clinicians or against these folks. I think we think very simply that target plus drug equals cure, and if the target is at a low frequency, it's not relevant, right? And I think that this approach, the work Todd's doing in these data sets, is going to change that. The low frequency target must be the one that you should be, and that might be the one that persists. So the approaches that we've taken, if you're looking for frequency-free or these mutations or some of these large translocations.
So you mentioned your R1 and the fact that you are taking these files and you place files on the sample. So what are the variables
Get a lot of the clinical data from the collaborators. So we will know in clinic we lack. We'll tell us exact information, but that's the goal is to get access to male, female, and all.
Because they're all part of a phase 3 study. It's an international study. There's quite a bit of phenotypic and demographic data available. What is ultimately going to be predictive and give us the best resolution? I think it remains to be tested and seen. I certainly think age, gender, ethnicity, those are easy things to pick out because we already know that there's big differences in those three parameters. And we don't do a good job of really discriminating for that currently. B617F is more common in men than women. So why is that? So
like environmental factors where they live or
I think so. You could look at what's in the germline. So not just cancer drivers, but there's polymorphisms in genes for the metabolism of chemotherapy, and so we sort of do that on sort of a spot basis. But you could imagine if you had somebody's genome or even their exome, and then you had on top of that their clonal data from an error-corrected approach. You could really pick the doses and the drugs and try to hit all the clones. I like it. I think we all like it, but I do worry that the public doesn't always think of what precision medicine is. Precision medicine is everybody's an N of is a family going to take my word for it if I say this is the best thing for you and nobody's ever had it before because you're completely unique? But trust me, it's going to be great. Are they going to go for that, or are they going to say forget it? How precise is precision? I don't know. So I don't think we want to outsmart ourselves. Let's keep it broad enough to be applicable to larger groups.
So basically, we are saying that we can predict tumor response when someone is treating.
Well, I think
Are there criteria that we should select and maybe use tests to
well, I mean, everybody has the right to know or not know. So, for people that would want to know what their clonal hematopoiesis looks like, I think you could offer it. If you had, I mean, I don't know. We're on the frontier here, right? I mean, so there's a lot of ways you could imagine if you found out that you had an 882 mutation, you should be screened more closely, or maybe there's some neoantigen that can be used for CAR T cells prophylactically. I mean, we couldn't do it today, but 5 years from now, 10 years from now. So, we won't get to those points unless we're actually generating prospective data to see, what can we do to prevent cancer before you actually, I think this is a little bit of that whole leukemia stem cell argument. You have 1 or 2 or 3 clones, and they've acquired these mutations. They're not rapidly dividing. I mean, 10 years later, they're still sitting at the same frequency, but they're there. And you get a mutation on top of that, and a mutation on top of that, and a mutation on top of that. Because, there's got to be a ton more mutation than what we're seeing. It just doesn't matter. It's happening everywhere. So, well, So you have a haploinsufficient allele on the other one, and now you have sort of a knockout effect or some other effect in that cell. I think that's a great question, and I hope that the well, I think, you could imagine that if you did an exome or a genome on somebody and you said, oh, look, 50% of their cells have a non-synonymous coding mutation in DNMT3A, and then they have, 1 in 1,000 of the hematopoietic progenitors have a different mutation in DNMT3A. Know that there's a 50% chance that that mutation is either on the same allele or the other allele. So, but the 882 acts like a dominant negative. You don't need to do this for that. To take out wild type function. So it's going to be different in every case.
Yeah, it will. I mean, certainly I'll like to take that.
It's a great puzzle.
For like, we're from genetic.
That single cell approach is the key for sure. The single cell approaches that we're looking at right now are customizable. They're sort of although it's highly heterogeneous, that's why it works. So we don't want to do that. So we have a targeted approach where we have some high fidelity identification with very specific sites cells that are constructed instead of a DNA molecule. And the cell can get in there and do the amplification, pull it out. So it's customizable.